Hello, my name is Alexandre Chicolet. I am postdoc at Sorbonne University, and today I'm going to walk you through the Plankton Genomics virtual lab, especially notebook 2 called Projecting Plankton Functional Gene Clusters that you will use for the topic Understanding the Ocean of Blue Cloud Hackathon. So I will go quite quickly through this video tutorial where I will explain you the different R pipelines and habitat modeling and mapping tools that you will find in this notebook. All the examples have already been run, so I spare you the waiting time. And yeah, when you arrive to the Blue Cloud gateway, you can access the data by clicking here, Demonstrator Plankton Genomics Demo 2. Here you will have a shortcut to notebook 2, and that's what's in it. So you will find a README document that will provide you with all the installation guidelines, where to find the files, the links for it, how to install them, and how to open your Jupyter instance in the Blue Cloud infrastructure. Also how to install the packages if you need. And so here you have a zip file with the technical documentation in it that you can find in this Word document as well. And you also find in this zip file all the codes that you need for running this notebook. So we can now open the Jupyter instance that we do when we arrive here. So once we unzipped everything, we have the following files. So service 1 IPY notebook is the technical documentation with an introduction, data sources, and the explanations for the inputs, outputs, and principle of the functions, as well as the code to run them. So everything is in here. I will go quickly through this. So for the data sources, you have environmental data that are climatologies. So here is the list of variables that are calculated with the average, standard deviation, etc. over this time periods. They are accessible through the data space Plankton Genomic datasets in this raster. And then we have genomic data that are the number of reads, so a proxy of abundance of genes per Tara Ocean stations. The genes are grouped in clusters of similar sequences, and they are also annotated by KEGG, pathway, and functions. So you can go on the KEGG pathway. Here you have the link websites. So we will take an example. Here you have all the KEGG pathways, for example, oxidative phosphorylation. Here you have the number corresponding to this KEGG, so the one that you will reference in the pipeline. And if you click on it, you have all the different functions, documentations, etc. about this pathway. So now that we know the data, framework structure is explained here. So first, we will load the different functions. Here, very important, the Blue Cloud directory is the directory where you unzipped your file, and data directory is where we find the environmental data. So these ones should not change. Once you have defined these ones, you can load config file, which will load all the function and libraries that you will need, as well as the functions related to each modeling step. So first query the database, then parameterizing the model, evaluate it, and do the bootstrap projections. For querying the database, we give the function which KEGG pathway we want, the size of the clusters that will be selected in it. So minimum size of 80 stations, a maximum number of genes, as well as the environmental metrics we want. So the average, standard deviation metrics, and distance and bathymetry variables. We can also say if we want relative abundance or not. once the query is done, you have, the size of your input for the model that are printed here. So the number of environmental features and cluster targets. Then we can train the model that uses multivariate gradient boosting, so that model all the clusters within cake pathway at once. You just give to this model an hyperparameter data frame, so the different parameters to test to have the best model, and it will select you the best hyperparameters here, number 1, so the red one, and the number of boosting round and the loss that correspond is printed. Once we selected our best model, we can evaluate it. So the model was trained on n-fold cross-validation, and we will evaluate on 1-fold that was kept for evaluation only. So to evaluate the model, we can evaluate, it's evaluated by the R-squared and the root mean square error on all the data. You can choose to do it by target if you want also, and you can print the variable importance also. So here you will have a series of graphic plots with the station number, the abundance, so the real one in black and the predicted one in gray for each target. So you can see the deviation of the model per stations. Once we are happy with the model evaluation, we can do the spatial projections. So here the projections are, we retrain a model and predict on all the data. So we will give, it will get the parameters corresponding to the validated model as well as the boosting rounds, and we just give the same environmental metric as we give to the first trained model. Keep in mind that this can take a little bit time, around 10 minutes for 1,000 boosting rounds, depending on which model was selected. And so this function will give you an output object, a raster, that we can then use for different plots. So among the plotting functions, we can plot the legend of the maps, that is a 2-scale legends. So the abundance is plotted with the colors, and the saturation will give you the standard deviation among every bootstrap run that we did. The maps are plotted with these functions, so 1 map per target cluster. Here we see that the relative abundance of each target is quite low, except cluster number 5, that is more tropical, has more relative abundance in the tropics, and cluster number 18 that has more abundance in the polar regions. So this is maps of genetic potential of some cluster that we modeled within this pathway. If you want to see which function do they correspond, we can refer to the query object of the first step. So query CC description. So here you have each cluster number and the main function they correspond. And finally, we can have also the correlation among, so the spatial correlation among each cluster. So here again, we can see that cluster 5, 11, and 18 are quite different than the others. I hope that this quick video helped you for understanding how this notebook works, and I'll be very happy to ask any question you might have here or within the Blue Cloud Hackathon. Thank you very much for understanding and listening this presentation.